Okay. So today will be our second lecture. So last time we kind of introduced the course and the expectation. So today we will start going into the actual content, the scientific content in computational biology and bioinformatics. So today we're going to start with let's say maybe not the most basic, but the most used application in bioinformatics, which is sequence alignment. So today we're going to talk about what is sequence alignment. Some of you may have used this before, but some of you this might be your first time. So why do we focus on this? Why sequence alignment as the first topic for the class? And we will learn about some algorithms, some strategies that we can use to perform sequence alignment. So when we talk about sequence alignment, we usually have two settings. Either a global sequence alignment or a local sequence alignment. So for a global alignment, so let's start from the bottom here. So for the global alignment, so the global here means that you have a target sequence in your database or in the genome of the species of interest, and you have some new sequence, maybe from experiment, maybe you sequence them yourself, or maybe you get it from another paper. So you have your query. What's called a query is what you have in your hand. The target sequence is what's in the database. And the basic question of sequence alignment is, does my sequence appear in the database? Does it exist in the database in some form? And this doesn't have to be a perfect match. So your sequence might have some mutation that can deviate from the genomes in the database. So the sequence alignment algorithm have to be able to not only recognize perfect matches, but also when there are some mismatches or there are some insertion and deletion. So the algorithm has to be able to calculate the similarity between your query sequence and the target sequence in the database. When it's a global alignment, you are expecting that your query sequence and the target sequence in the database are of the same length. So you expect that you have the whole sequence, basically. So global here means that the alignment expect the start of your sequence to be roughly similar to the start of the target sequence, and the end of your sequence will be similar to the end of the target sequence. You can see that the alignment can occur throughout the whole sequence. But for local alignment, sometimes you clone only part of the gene. You don't have the whole gene, but you might clone like the 5' region of the gene. So you can still search the database and ask, does my sequence segment, just a fragment of it, does it appear in the database? So this will be a local alignment. So the algorithm will assume that your sequence, your query sequence, will be a subset, will be just a part of the whole genome of protein or gene sequence. So this is usually the two settings that we will talk about. And sequence alignment has been used for, sorry, the sequence is long, has been used for a long time. So since 1970s, when people start to develop the first DNA sequencing technique called Sanger sequencing. And we will talk about the actual techniques and the evolution of DNA sequencing techniques next lectures. And you might be surprised that some of you might have downloaded genomics data from GenBank before. So this is the US database for genomics data. GenBank has been established since 1980s. So people start to be able to sequence DNA and protein and gene. And then next, once you have some sequence, you want to search and compare if your protein have similar sequence as another lab's protein, so that you can infer that, oh, we are studying the same thing, we are studying the same protein. Or you want to know if the gene that you got maybe from some bacteria, which species does it come from? You want to compare your gene sequence to some genomes of bacteria in the database and ask this question. So sequence alignment has been, let's say, almost probably the first bioinformatics technique that people start using. And if you go back and look at the paper for the BLAST, Basic Local Alignment Search Tool, or BLAST BLAST, this has been invented since 1990. So people from the computer science background start to help the biologists analyze their DNA sequence and protein sequence data from very long time ago. And you can imagine that back in the day, if you imagine 30, 40 years ago, like biologists didn't yet have the high throughput sequencing. They can only experiment kind of one cell culture at a time, one bacteria at a time. So they might find kind of new bacteria or new cell culture that have a certain function. In the lab, you might find certain phenotype that are interesting, like the cells might be able to produce some compound. And then you want to know like protein or which gene in the cells that are responsible for that function for producing that compound. So you might be able to narrow down the gene, like you might extract some of the genes, and then you can test in the lab that, okay, if I insert this gene into a new bacteria, into a new cells, then you can reproduce that function. You can create that compound. But once you know the gene, next, you would want to know the DNA sequence of the gene so that we can maybe clone it, we can edit it. So back in the day, people start to have DNA sequences, and then they want to know if the new gene that they found, is it new? Or is it something that people in other labs have found already? So they want to search the new DNA sequence against the database. Does this gene or does other gene with similar sequence appear before in some prior study? And from sequence similarity, we can infer many things. So the key of sequence alignment is that once we can align the sequence, we can tell whether sequence A and B are similar. In terms of biology, if we have two sequences that are similar, then we can do a lot of prediction. And the similarity between sequence, the technical term is sequence homology. So you have some similarity or homology between the sequence. And the reason why we can infer or we can predict a lot of things, a lot of biological information from similarity at the sequence level is because evolution, like the process of evolution, occurs at the sequence level. You have mutation in the DNA, which then got translated into proteins. So that means that if you have gene or protein that were derived from the same ancestor, like from the same original gene, then they start with the same sequence and then they evolved. So at the end, like in different species, genes with the same function or genes from the same family tend to have similar sequence. So that's because of evolution. And we also know that from evolution that if you introduce mutation to gene and protein, in most of the cases, you don't change the function, right? Because many nucleotide or amino acid position, you can change them and they doesn't change the protein 3D structure, they doesn't change the protein function. So by this observation, we can kind of assume that if we find two DNA sequences that are similar, we can imagine that, oh, they might derive from the same origin, right? They might derive from the same gene in the ancestor, and they became different gene in different species through evolution. So by this process, we can assume that, okay, if we find genes like this, then they might have the same function because in the beginning, they have the same function. And after a certain evolutionary period, it's more likely that they retain the same function more than that they adopt different abilities. So if we see similarity in sequence, we can infer similarity in function. And in later slides, in later lectures of this course, you will find a more advanced way to annotate gene and protein function by using sequence similarity. But for now, let's focus on the idea that if you can find similarities in sequence, then you can make prediction about the gene and protein function. Another factor is you can also predict 3D structure of a protein based on sequence similarity. Because we know that protein that originated from the same ancestor, they can accumulate different mutations, but usually the 3D structure and the functions remain the same. So this principle also helps kind of biochemists when they want to model the 3D structure for new proteins. Instead of having to calculate the new protein structure from scratch, they can take the amino acid sequence of the new protein, align it to the amino acid sequence of a known protein with known 3D structure. and they can predict that the secondary structure, the tertiary structure, the quaternary structure might be the same because these proteins are similar. So in this figure, I show you that we have two proteins, one in blue and one in red. We can align the sequence, the amino acid sequence, and see some similarity in this position. And we can infer that the structure for the red protein should be similar to the structure for the blue protein because of this similarity in sequence. And it's quite good if you have protein for the same family or protein that are not too far apart in terms of the evolution. Another application for sequence alignment is for those of you who do wet lab. Right? When you do wet lab, you have to design, let's say, CRISPR guide RNA, or you might have to design PCR primer. So when you design primer or guide RNA, your goal is to be specific. Like you want your CRISPR kit to target a certain gene, a certain part of the genome. So you want to make sure that the sequence that you design, if you align it to the genome, if you align this to the genome, you should get very specific hit to the genes or to the region that you want to target and nowhere else. Right? If your probe, if your guide RNA can target 5 different regions in the genome, then you don't know if your CRISPR assay will target position 1, 2, 3, 4, or 5. Like you want to make sure that it's specific. And this can be done with sequence alignment. If it's aligned at one position, but not aligned with other position, then you know that it's specific to the where it's aligned to. Okay. So for those of you who are interested in evolutionary analysis, and we will talk about evolution in a few lectures, we use sequence alignment a lot to infer evolutionary information across species, because we know that every species originated from the same ancestor. So the same gene will accumulate different mutations. So if you can align genes together, you can tell that this mouse gene is similar to this human gene. Then you can infer that, oh, this mouse gene and this human gene are probably the same gene, originated from the same ancestor of all mammals. Okay. Now we'll talk more about this when we learn about evolution. But just want to summarize that with sequence alignment, by being able to compare similarity between two sequences, you can infer evolutionary relationship. You can identify the species of origin, like you might have some DNA fragment from a patient's blood, and you want to know if this comes from pathogen or does this come from a harmless bacteria. You can do this with sequence alignment by aligning the fragment you get, the DNA that you have, against a database of microorganisms. You can have the answer, like which species that this comes from. And you can use sequence similarity to predict the structure for the protein. We'll talk more about this in the protein structure modeling session, but here's another application of sequence alignment. Okay, so now I hope that all of you are convinced that sequence alignment is a powerful tool that we have in computational biology. So now we want to look at the algorithms or the strategies that computer scientists can use to perform sequence alignment. So let's start simple. So let's say we have two sequences, two DNA sequence, the top and the bottom here, and I help everyone by highlighting the region in bold here. These are perfect match between the two. Okay? So if I ask you to align the top and the bottom sequence, okay, some of you might have an intuition that the alignment that would be the best one probably have to align these two bolded regions together because they are perfect match, right? And they consist of more than 10 kind of nucleotides. So the best alignment that we can make should align these two regions together. And the front and the back might be different, right? But the central part of the alignment probably have to be like this. So this is an intuition that computer scientists also think about when designing algorithms. And this is what they use. So let's think about the same idea. This idea that if we have a region like with the perfect match, so the DNA sequence in these regions are identical. How likely it is? How likely it is in terms of statistics? So let's say we have an input sequence of length 300, so 300 nucleotide, and let's say you are resequencing the genome. So you are studying human cells, you sequence the gene, it might have some mutation. You are aligning this, you are comparing this to a human genome database. So you expect that your sequence is probably very similar to whatever is in the database because you are sequencing human genome again. So in this case, you might expect 95% similarity. So since we expect 95% similarity, we are going to expect from 300 nucleotide, we are going to expect 285 position to be identical. And 15 position might be different, maybe because of mutation, maybe because of sequencing error. But we only expect 15 position out of 300 to be different. Okay? So let's think if we have 300 position, and we have 15 positions that do not match. So let's imagine that we have 300 characters, and M stand for matches, and E stand for errors. So errors mean a mismatch. So we have 285 M's and only 15 E's. So at best, if you want to place E into this sequence, so that the let's say the length of consecutive M's, okay? The length of consecutive M's are as short as possible. The best you can do is putting all the errors evenly in the sequence, and you divide the sequence of length 300 into 18, into kind of stretch of 18 M's, separated by E. Okay? So this is the best you can do. If you move the E in any other location, you get a longer stretch of M's. So in this case, because you know beforehand that you're going to get a hit or you're going to have an answer with 95% similarity, you can assure that whatever result you have is going to have 18 matches in a row. And this is because of a branch of mathematics called combinatorics. But anyway, you can all see that we can guarantee some because we have very few errors. And this is a strategy that BLAST and sequence alignment algorithm use. So if you assume that you have 95% similarity between your input and the database, actually when you run BLAST on NCBI, they're gonna assume 28 matches in a row because you should have very few errors and you should have a lot of matches. So this is a way to kind of, instead of having to consider every possibility, you can look for a position with perfect matches first. So let's see how the algorithm works. Okay, I think I changed the order of the slides. Okay, but anyway, so up to this point, like we have an intuition that if we have two sequences and we have certain expectation about their similarity, then we have some expectation about the length of perfect matches, and this can be calculated. And computer algorithms can exploit this relationship to say that if any answer were to be kind of produced, that answer should have a certain number of matches in a row, and this can be a criteria that can speed up the search. So this is one idea. Another idea that I want to introduce is called dynamic programming. So for those of you not in computer science, this might be a new term, but this is something that you all are very familiar about. And let's think about this in terms of a game. So let's say we have a pile of 20 logs, or you can think of 20 coins, 20 balls. You have 20 objects, and we have two players taking turns, so one player at a time. And what each player can do is remove at least at either 1 or 2 logs from the path. And the goal is whoever removes the last logs win. So you want to be the last person to remove the log from this path and make it 0. So the question in mathematics and computer science is, can we determine who the winner is? If I give you 20 logs and you play this game, right, it might take a while for you to figure out kind of who will win, who will have the best strategy. But dynamic programming is the idea that if I change the number of logs from 20 to 1, this is very easy. If you have only 1 log, then the first player just take that 1 log, right? And that player will win. So the first player will win if we have only 1 log. If we have 2, then the first player still win because he or she can just take both of the logs, and then that person will be the last to remove. If we have 3, right, now this becomes interesting because the first player cannot finish taking all the logs in one turn. Okay? So because he or she can only take 1 or 2 logs, like if he takes 1, then the other player will take 2. If he or she takes 2, then the other player will take 1. So whatever the first player did, the second player will always win because he or she can take the remaining logs. Now, if we have 4, then the first player win. And this is because the first player can take out 1 log and then send 3 logs to the other player. And we know beforehand that if anyone is faced with 3 logs, that person loses, the other person win. So with 4 logs, like the first player can take out 1 log, send the 3 logs to the second player, and so the second player will lose. So with 4 logs, the first player wins. Similarly for 5, if we have 5, the first player takes out 2 logs, right? And the second player is again faced with 3 logs, and that person will lose. When we go to 6, right? Whatever the first player does, taking 1 or taking 2, the next players will be winning because that's he or she has a strategy for when there are 4 or 5 in a log. And we can repeat this pattern. It will be first player, second player, first player, second player. And this will repeat because the same argument applies everywhere. So now we can tell that with 20 logs, the first player wins. So now that you know this technique, this card game will no longer interest you because it's fully solved by mathematics and computer science. But this algorithm, the reason why it's powerful is because if you have something the problem that's very complex, like you have a lot of logs, you have a very long nucleotide sequence, right? It's difficult to find the best alignment. It's difficult to find the answer. But if you simplify it into smaller problem with fewer logs, with shorter DNA sequence, then you can use answer from the smaller problem to answer the larger problem. So let's take another example that will bring you closer to sequence alignment. So this is just kind of one-dimensional game, like you have 20 logs and you take the logs out. Let's consider a 2-dimensional game. So we have starting point at A and then the end point at B. Right? One of the classic question is how many ways can you travel from A to B using only the right and the up direction? How many ways you can kind of walk from A to B? And for mathematics students, they will answer 35 ways right away because this is a classic problem. But we are going to use dynamic programming techniques to answer this problem. And we'll see that it works all the time. So let's say we start at A, right? So the number of ways that we can go to A is only 1 because that's the starting point. Right? And the only way that we can visit the position above A, there's only 1 way to go up, 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 and up. There's only 1 way to visit all the node or the point to the right of A, which is to go right, go right, go right, go right. You only have 1 way. You have no variation. But let's say, how many ways can we visit the position to the northeast of A? There are two ways. You can go up and then right, or you can go right and then up. And you notice that this 2 here is actually 1 plus 1. Right? So you have one way of reaching this point from the right, from the left, and you have one way of reaching this point from the bottom. And each of them has one way for you to arrive at that location. The next layers have 3 ways, right? Because you have one way to reach this point from the left, and you have one way to reach this point from the bottom, 2 ways to reach this point from the bottom. So you have a total of 3 ways. So this is also the same. You have 2 ways of reaching it from the left and 1 way of reaching it from the bottom. So you can add 1 and 2 and get 3. And you can keep doing this. We have 3 ways of reaching the number 6 here, we have 3 ways of reaching this point from the left, and we have 3 ways of reaching this point from the bottom. So we have 6 ways in total to reach this position. And we can keep doing this until we get the final answer, 35. And for those of you who remember, this is a Pascal triangle, the number that appears in the Pascal triangle. Anyway, so you can see that I can expand the grid. Like this is only 3 by 4. Like, it's really simple. You might say that I can just count the number of ways. But if I expand it to, let's say, 30 by 40 grids, then without this process, it will be difficult to tabulate the number of ways to go from A to B. And let me show you another example. Let's make it more complicated. Let's say we have 2 aliens waiting for us at 2 positions, and we want to avoid the alien. So we want to go from A to B without running into the alien. I should be able to come up with better obstacle in terms of biology, but I'm not sure. So let's use alien for now. So again, we know that starting at A, there's only one way to go up and up, only one way to go right, and right. And we have 2 ways of reaching this point, one from the left and one from the bottom. We have 3 ways of reaching this point, one from the left and one from the bottom. Now it becomes more interesting because we have aliens. The next layer doesn't follow the same pattern as before, right? Because we cannot go through the alien. So this point that I just circled, this point, we only have one way of reaching it by going right, then up. There's no other way. This point, we only have 3 ways, which is to go to this point first, and then you take the right here. So you see that this dynamic programming techniques works even when you introduce obstacle onto the grid as well. So at the end, you can sum all the numbers and then get 18. So this process is very powerful because you can solve very complex problems by just working out the simpler one beforehand. So how does this help us with sequence alignment? So let's see the third example for actual sequence alignment. So let's say we want to align sequence AGCT and ATGCT, very similar. So the idea is we start from the top left corner, and this is where we don't align anything yet, right? And the score is 0 because we don't align anything yet. Let's say we have an agreement beforehand that if we have a match, so A to A, B, T to T, G to G, C to C, we're going to give +1 score because it's a match, it's good. If we have a mismatch, A to T, A to G, A to C, for example, we're going to deduct 1 point because it's a mismatch. And if we need to introduce gap, which represent insertion and deletion, we're going to take out 2 points because you are going to introduce some evolutionary process in there. Let's see how we do the alignment using this kind of scoring agreement. So from 0 from the top, right? If we go to the right, this means that we align A from the top sequence against gap. So this means insertion or deletion. If we further align T to the gap, this means we have 2 insertion, 2 deletion. G also 3 insertion, 3 deletion. So we can fill the score, it will be -2, -2, -2, -2. The same for the column, for the first column. If we align A in the left sequence to the gap, that means we add insertion or deletion. So it's -2. Another insertion deletion, -4. Another insertion deletion, -6. So the score can accumulate. The numbers that are shown in this table is the accumulated score. If you start from the top left corner to that cell, to that position. So let's say, can we do better? We say that, okay, since the first sequence, both sequence starts with A, okay, we can align them together. And this gives us a match. So we have A +1 score. But now we have a choice, right? Because T and G do not match. So we have either introducing a deletion, insertion deletion. So we take A -2 and put the insertion with deletion there. Or we can go down, introduce insertion deletion on another sequence, and also again take A -2 score. Or we can put T and G together, and this will be a mismatch. So the score will be back to 0. We can repeat this process of considering what happens if we go to certain cells from certain direction, and we can fill out the score. So this also becomes 0 because we have the score was -1, right? And we have G and G being paired together, so we got a +1. So this again also becomes 0. And then we can fill out the process. Good. Notice that the arrow tells us how to reach that position and achieve the best score. Right? Because there are ways you can reach a certain cell by using a lot of deletion and a lot of insertion, but those give you very poor score. So these arrows can keep track of how can we reach this number 1, how can we reach this 0, how can we reach this 0 by following the insertion, deletion, mismatch, or match. Okay. So we can keep filling this, right? We consider from, let's say from T and G here, so the score is 0. And we can either introduce insertion deletion and get -2. Or we can put G and C together and get a mismatch, -1. So there's no way to get positive score from this position. So at the end of the day, we're going to figure out that the best way to reach the bottom right corner, so we want to go from the top left down to the bottom right. And the best way is to follow the green arrow to put A together, A and A together into a mesh, put T as an insertion deletion, and then align the G together, align the C together, and then at the end, align the T together. So this is the result that you have, going from the top left to the bottom right. And this is the alignment that we get. So A align to A, T become an insertion, G, C and T are aligned together. So this arrow, the diagonal arrow, tells you that we take the sequence from the top and the left and align them. If we have a horizontal or vertical arrows, this means we introduce insertion deletion. So in this case, we have one mesh, one insertion deletion, second mesh, third mesh, and then fourth mesh. And the total score at the end is +2. And this is the best alignment that we have, because from the top left to the bottom right, this is the let's say the path that give us the best kind of score. And this is for global sequence alignment. To remind you, so global sequence alignment, we assume that both sequence here must be aligned from end to end. Let's look at another example. In this case, it's the same sequence as before, but we are considering local alignment. So to remind you, so local alignment means that the shorter sequence doesn't have to align to the whole sequence for the longer one. It can be just part of it. So in this case, you can see that there's no cells with negative score. Everything becomes 0. And then you have 1 and 1, 2, 3. If you compare this to before, we have 1, 2, but the rest are negatives or 0. And the reason is because for global alignment, we have to go from the top left to the bottom right to align the sequences from one end to the other end. So even if we have to get negative score from gap or mismatch, we have to take it in order to align from end to end. But for local alignment, like if you cannot get a good alignment, you can just skip that region. So, for example, this is the result that we have. So basically, this numbers tells us that the A from the second sequence and A and T from the first sequence are not part of the alignment because you don't have any way to align them and get positive score. Instead, we found that considering just the G, C, and T give us a positive 3 because these 3 positions are aligned together. So basically, the idea for local alignment is if we cannot create a positive score from the alignment, we can start over. We can discard some of the sequence and align the rest. So this is the idea. That is actually used inside all the tools that you have been using. So to rephrase in terms of wording, so dynamic programming for sequence alignment, the way that it kind of make the problem simpler is if we consider sequence alignment between these two sequences, let's start with the orange T and start with the green T. That somehow sounds like drinks. Okay. So the idea is you have 3 choices. You can either align the T together, or you can add insertion on one of the sequence. You only have 3 options. Okay. If we go back to this example, notice that we only have 3 types of arrow: horizontal arrow, diagonal arrow, and vertical arrow. So you will have 3 options at each step. And these 3 options means if it's a diagonal arrow, you are aligning the nucleotide together. If it's a horizontal, then you add insertion deletion to one sequence. If it's vertical, then you add insertion deletion to another sequence. And once you do this, your sequence becomes shorter. So the best alignment for the original question is one of these 3, plus the best alignment for the sequence that are slightly shorter, one nucleotide shorter. So you can keep doing this. You can take TCATA and CGTA, and then you can consider the same thing. There are 3 ways that you can align these two, and the sequence will be shortened. So this is how dynamic programming is applied to sequence alignment. So now that we know the algorithms, we know the steps. Next would be the question of how did we come up with these numbers 1, -1, and -2? How do we kind of define this, and how do we make sure that these numbers 1, -1, and -2 will lead us to a correct alignment? Does this correspond anywhere to biology? So that will be the next topics. We want to consider the score that we are going to use for the matches, for the mismatches, and for the gaps. How do computer scientists kind of define this for us? So the scoring of sequence alignment, if you go to NCBI, and you will have a chance to go to NCBI website to perform blast, there will be options for the scoring parameters, and this is for nucleotide alignment. And they will let you specify the match and mismatch score. Like in this case, it's either 1 versus -2 or 2 versus -3. And they will also allow you to select how would you score the insertion deletion or these batches. There are 3 choices for the insertion deletion. There's a constant model. So no matter how many insertion deletion you put in, you're going to get penalized the same way. There is a linear model where if you insert longer deletion, like 3 dashes, you're going to get 3 times the penalty. So it's a penalty cost multiplied by the length. And you're going to have something called F5 cost. And F5 is also like a linear function. So you have a constant that if you introduce an insertion deletion, you're going to get a fixed penalty. And then once you expand that insertion deletion further to be 2 dashes, 3 dashes, 4 dashes, you're going to get additional penalty that got multiplied by the length. So you can see that F5 is a combination between constant and linear. So there's a constant part, which is the existence of the deletion. And there's a linear part, which is the extension of the insertion and deletion. And let's say we have this alignment. between these two sequences. There are 4 matches and 3 insertion deletion. So if you use the first scoring criteria, it's going to be plus 6, like 6 matches, and then minus 3, because it's a linear score. We only minus each insertion deletion got minus 1, and there are 3 of them, so it's minus 3. But if we use the second model, for each match we got plus 2, so it's plus 2 times 6, 12, and then we have 3 gaps, so we pay the price with 5, 5 being the existence of this gap, this insertion deletion, and then for each position we get minus 2. So this is the score that we get. So different score given the same alignment, different score parameter, different score kind of scheme will give you different result. Plus 3, plus 1. So let's see what this means. Let's think about the match and mismatch score first. Why did they give you a default of plus 1 minus 2 and then plus 2 minus 3? So what do this mean? So let's say we look at the match to mismatch of plus 1 versus minus 2. This means that if you have one mismatch with 2 matches, one mismatch and 2 matches, you have no score. You get nothing, because one mismatch cancel out the 2 matches. So this means that the result that you have is going to have very few mismatch, because for each mismatch you're going to have at least 2 match to cancel it out. So every answer that you get is going to have very few mismatches. And this is good for finding DNA from closely related species. You expect very high similarity. You don't want any mismatches in the answer. But let's say you use a match to mismatch of plus 2 and minus 3. Right? The same situation, if you have one mismatch and you have 2 matches. In this case, you gain a net of plus 1 score. So this scoring scheme allows more mismatch into the answer, because you can offset 2 mismatch by 2 matches and you get plus 1. Before, in the top setting here, you get nothing if you have 1 mismatch and 2 matches. But in the bottom, if you have the same situation, you actually get more. You get an increase in the score. So the bottom settings here will allow more mismatch into the answer. So it's better for finding more distant species, or if you expect your sequence to be less similar to the database. Okay. Next, let's interpret what the gap kind of score means. If you have a constant gap score, that means that you allow insertion deletion to be of any length. Okay? You imagine that if there is a deletion in terms of biology, that deletion occurs on the DNA, it might delete 1 position, 2 position, 3 position. It doesn't, in terms of biological mechanism, it might not cost more energy to delete 3 versus 4 versus 5. It's just a single reaction. So you can imagine that under that assumption, a constant model might be appropriate, because you assume that an insertion is an insertion. It can insert a whole gene, it can insert half a gene, it's still one insertion. That way you think of insertion and deletion as a constant, just it appearing on. But if you think that there is a core, the longer the deletion you have is less likely. So let's say you have kind of closely related species, and you think that the evolution will try to keep the protein to be the same or the gene to be the same. If you have longer and longer insertion and deletion, that means the sequence are further and further different. So in this case, you might want to penalize. You want answer that doesn't have very long insertion and deletion. So you start to use either the linear model or the affine model. And this kind of depends on your assumption. It doesn't, I want you to think about kind of the meanings in terms of biology before kind of choosing the scoring scheme. Okay. So let's see how these scoring scheme and techniques were implemented in BLAST. So if you go to the NCBI's kind of BLAST website to search, it will ask you for two essential things: your query sequence, like the sequence that you have, and the database. What do you want to search against? So you can search against kind of specific database like human genome and human transcript, or mouse genome and mouse transcript. So these are very popular for people who do study in animal, in human and animal. But you can also search other things. You can look at kind of different taxonomy ID. If you are studying kind of DNA from human blood, like you might want to study pathogen specifically, you can make that choices here. And then the most important part, because we are computational biology courses, we want to pay attention to the actual algorithms. So the default for nucleotide blast on NCBI is called megablast. And megablast, they say that it's for highly similar sequences. And if you click on the description, it will tell you that megablast is intended for when your sequence is expected to be 95% or more similar to the database. So you basically want something that's very similar. And these are reflected in the settings. So you can, as a user, you can just select megablast or discontiguous megablast or blast it. And it will tell you that you can get highly similar sequence, you can get somewhat similar sequence. That doesn't really mean much in terms of what it actually does. But if you look at the actual settings underneath, you will find that for megablast, word size here means that any answer that it produces is going to have a region of 28 nucleotide that is perfectly matched to your query sequence. So this is the first idea that we talked about. Okay? If we have a certain length of query and we have a certain expectation of sequence identity of matches, then we can expect a certain range, a certain length of perfect matches. For megablast, it's 28 nucleotide perfectly matched. And it used a mismatch score of +1 -2. So this means that we talked before, this means that we don't want to introduce mismatch to the answer. For BLAST-N, which they say that is for a somewhat similar sequence, because we expect more differences, the length of perfect matches reduced from 28 to 11. So we don't expect such long perfect matches, we expect shorter matches. And the match to mismatch scoring is now +2 and -3. So the algorithm now allow more mismatches into the result. So they make this preset for you, and they have like a kind of description in wording, right? But if now that we understand the algorithms, you can go back and look inside the actual parameters, and you understand them, and you understand what the algorithms is trying to do. Megablast is trying to get a sequence that have a lot of matches in both in terms of the length of perfect matches and also in terms of the ratio between match and mismatch score. On the other hand, BLAST-N kind of reduce the length of expected match from 28 to 11, and it gives more weight to and it gives more possibility to introduce mismatches. Okay, so this is how they kind of achieve the highly similar sequence and somewhat similar sequence in the description by setting the parameters to be corresponding to that target, to that goal. So let's compare what you get. Right? If you put in the same sequence and you run megablast or you run BLAST-N. With megablast, you will get very few hits because all the answers must be very similar. With BLAST-N, you're going to get more possibilities. So this depends on your application. If you know that you are targeting, you are designing CRISPR or primer, and you are targeting the human genome, maybe you can focus on megablast because you want to be very specific to that region. But if you have a sequence, maybe from a virus, maybe from some species, and you want to know like which species or which kind of taxonomy class does the sequence come from, you might want to try BLAST-N. because you have no idea. Right? There might be a similar genome in the database, or there might not be a similar genome in the database. So you can use BLAST-N to get more possibility, to get more coverage out of the result. Okay. So when you look at the BLAST result, there are three numbers that are very important. I highlight them with arrows here. The first one is query coverage. So let's say you put in a sequence of length 100 nucleotides. Query coverage will tell you what percentage of your input sequence is part of the alignment. If you look at the color bar here, the red bars, this represent the first hit. The purple bar represent the second hit. And you will see that the first bar has a cover, has a much larger score. And when we look at the bottom here, we see that the coverage, the query coverage for the first hit is 100%. So the sequence that you put in is matched to the human genome for the entire length, 100%. But the second position in the human genome that your sequence can match to only cover 43% of your sequence. So less than half of your sequence matches. So query coverage can tell you how confident you are that the match that you get from BLAST is correct. If you have 100% coverage, then basically that's the perfect match for the whole thing. Not perfect, but it's the match to the whole sequence. But 43%, like what happened to the other 57? Right? Does that mean that it's related to human genes or not? Because more than half doesn't match. So that tells you like how long of your sequence match to the database. The second component is the identity. For the region that match, like 100% or 43%, how well does it match? How many mismatch? How many matches? So you can see that for the first hit is 100% coverage and 100% identity. So this means that every nucleotide position matched to the genome. So basically you are copying part of the genome in your design. But the second hit only cover 43% of your sequence, so less than half. But within that 43%, the match is 84%. So within that 43% of your sequence, most of them are matches. Only 16% is mismatches. So these two numbers can give you an idea of how well your input match to the database. But to summarize everything and allow you to consider just one score, for BLAST, it will give you what's called an E-value. E-value here, in this column, this tells you the expected number of hits that you would get with the same or higher score by chance. So this is similar to p-value. We want to know, once we have, let's say we have like 43% coverage and 84% identity. So this is just like describing the result. But in terms of statistics, we want to know if this is significant or not. So E-value kind of stand in for the statistics for sequence alignment result. Usually people cut off this as 1 to the negative 1 to the 1 times 10 to the minus 5. So it's 0.000001. So it's very low. So that means that if the chance of getting a hit in the genome, even though your sequence does come from that genome, should be very small, less than 1 in 10 to the 5. That you will see this by chance, similar to p-value. But let's think about how they derive E-value. We have sequence alignment. How do we turn that into a statistics? Because I think from what we learned so far in statistics class in university, I don't believe anyone has seen a statistical model for sequence alignment. So let's take this chance to analyze how does people define the E-value for sequence alignment result. And our setting is that we have an input sequence. So this is your query. You have an input sequence of certain length. So n is just a number. And your database, like human genome, is 3 times 10 to the 9 nucleotide. So that's the capital N. That's the size of the database. Let's say we have a BLAST result with score S, okay? S is just a score. What I want to discuss with you is that the e-value or the statistics of the sequence alignment result should be proportional to n times m times e to the minus some constant times the score. You don't have to remember this, but we want to be able to discuss why it's in this form. Let's think about this. So let's say we have okay, let's say we have your input sequence of certain length. Okay? Let's say you input another sequence that's twice as long. So your sequence length now becomes 2n. Let's say you put the same sequence twice. So the black and the gray region here, just imagine that you can copy paste your sequence twice. So whatever blast alignment you get from the first portion, the black part, you're going to get also in the second part because you copy and paste the same sequence. So this means that if your input sequence become twice as long, you can expect to have twice as many hits, many results, many answers with the same score. Because these red, green, and blue, these are the hit with score S or higher. So if you have twice as long input sequence or query sequence, you expect to have twice as many matches. So that's why the e-value, which is the expected number of hits, scale linearly with the length of your input sequence. In the same consideration, if we think about the database, the genome, if we scale the database by twice, like we copy and paste the database twice, then we also expect to have twice as many results. So this is also why the e-value scale linearly with the length of your database. So if you have a larger genome, you expect to have more hits based on the size of the genome. The last one is maybe more tricky. So let's say we have a matches with score of 2s, right? 2 times the score. Recall the dynamic programming, the example that we have. The score that we get from sequence alignment gets added, right? As you align more and more position, you plus or minus the score. Okay? So the score adds up as you have longer and longer alignment. So you can think of a match with score 2s as 2 consecutive matches, each with score s. Because let's say you start with 0 score, like you at a certain point in the alignment, the score becomes s. And then after a certain point, you plus s and you get 2s. So basically you have 2 segments of the match of the alignment, each producing a score plus s. And together you get 2s. And the probability that you get the score of s twice in a row, and these are 2 regions, 2 different parts of your sequence, each produce a score of s. So by, not sure if I can use probability here, but hopefully you can imagine that if you have a certain probability of getting a score of s in the green region and a certain probability of getting a score of s in the gray region, the probability that you get a score of 2s together next to each other is you have to multiply the probability. So you can think of this as if you roll a dice, and the probability of getting this score is the probability of getting a 1 out of your dice. If you were to roll the dice twice in a row and get 1 and 1, it's the probability of getting 1 times the probability of getting another 1. So it's multiplicative. So this kind of shows that if you increase the score by 2-fold, your score actually becomes exponential because you are multiplying the smaller score, not plus them together, not add them together. So the e-value in terms of the score of the alignment doesn't scale linearly, it scales exponentially. So these 3 kind of situations can illustrate why the e-value for the sequence alignment have to be this form. The length of the input times the length of the database times exponential of the alignment score. that we get from the dynamic programming. So this is how kind of they develop the eval that we use in BLAST. Okay. Another, we haven't reviewed the probability yet. We will do this kind of rigorously in the latter half of the course. But I'm pretty sure that all of you have heard of Poisson distribution before. So Poisson distribution describe if you have a certain event, a certain happening, a certain occurring within a certain confine of space or within a certain period of time. And in this case, we can think of the event of having a alignment with a score of S or higher as the event, right? As the happening, as the occurrence. And the scope is the genome. So we think of the genome as being our confined space, and we can find certain alignment in this genome with a certain score. So this can be described as a Poisson distribution. And we will talk more about this in the later session. So for now, I just want you to know that we can actually model sequence alignment score, the e-value, using a distribution that all of you have seen before. Basically, the statistics that we use in bioinformatics and computational biology is not that scary. It's something that you already know, but applied it in a different setting. Yep. Another consideration that I want you to be aware of is when we think about score from the sequence alignment, right, and we want to analyze the statistical significance for the score. One thing that affects the statistical significance is the composition of the nucleotide or protein that you are considering. And this is called low complexity region. So let's say you have a segment of your genome that have C's and G, like these are CG islands. You know that these have certain regulatory roles in the genome. In this region, it's very easy for you to find good matches because you will have C and G. Right? Imagine if you have A, T, C, and G, it's only 1 in 4 that you can get a match. But if your sequence consists of only C and G, it's 1/2. Right? You either see, you either get a match or you get a mismatch. So G and so when you have a region that have very low complexity, so very skewed nucleotide distribution, you don't want to consider this in your scoring because it will bias the score toward higher numbers. So in BLAST, there's a default option that it will be checked. Say it will mask, so it will remove basically any region with low complexity from the calculation. So your result will not be biased by these patterns. Something very important is on by default because most of the time you need it, but it's hidden. So I want you to kind of know that there are these considerations that are inside the algorithm. Okay. So those are for nucleotide alignment. Okay. For nucleotide alignment, the scoring is simple because between A, T, C, and G, mutation can change anything to anything. You can go from A to T, you can go from A to G, it doesn't really matter much. But when we want to align protein sequence, okay, we can go back to your biochemistry class. Like we know that the 20 amino acids, actually there are more now, but the major 20 amino acids have different side chains. And each of them have different chemical and physical properties. Some of the amino acid has a charged side chain, positive charge or negative charge. Some amino acids have a hydrocarbon side chain, like only hydrocarbon. Some even have large, bulky side chain like aromatic rings. And there are some special cases like cysteine, which has the sulfur, so it can form a disulfide bond. And proline, which is structurally different, is angled, not linear. So these amino acids have different structural and functional meaning, unlike nucleotide where A, T, C, and G by cell doesn't really have any functional or doesn't have many functional or structural meaning. So for amino acids, we can group them based on function, and we observe that from evolution that if you have a mutation that doesn't change the side chain much, it becomes a different amino acid, but it's still hydrocarbon, or it's still charged, or it's still bulky, nature will tolerate. So it's a mutation that doesn't cause problem. So we can accumulate these mutations. So now we need to be able to determine the scoring between all 20 to 20 amino acids in such a way that if it's a similar amino acid with similar function that nature can tolerate, we should give it positive score. But if it's a change in amino acid that totally change the function on the side chain, we should probably give it negative score. And how can we do this? We can, so in the beginning, people have no idea how to do this because we don't know how the protein evolved yet. But after a certain point, when people start to collect more and more protein and gene sequences, we start to know different sequences from the same protein family. So the same protein, but from different organisms. We study yeast, we study fruit fly, we study C. elegans, human, mouse. Once we know the same protein from many species, we start to be able to recognize which amino acid can turn into a different amino acid based on the actual data in nature. So people use that information to construct this kind of metrics that says to us that if a glutamate change into an aspartate, it's still acid, so the score is +2, it's okay. But if a glutamate is changed into a cysteine, this is not okay. You totally change the side chain and the score becomes -4. So these metrics come from the actual patterns in evolution, whether a certain residue, a certain amino acid can turn into another amino acid or not. And you see names like BLOSUM, which stands for block substitution matrix. So it's just a matrix of the score between two amino acids. And you will see numbers following, like BLOSUM 45, BLOSUM 80, BLOSUM 60. And this tells you when they construct this matrix, like for BLOSUM 45, this is constructed using proteins with at least 45% conservation. So that means that if you see BLOSUM 45 and BLOSUM 60, like the BLOSUM 60 would be constructed using more conserved protein, more than 60% conservation. 45 would be less conserved, more than 45% conservation. So different level of conservation will give you different kind of implication in terms of evolution. So this is one way to calculate the score for the substitution or the match or mismatches between amino acids. The second way, and this is more theoretical, so for those of you who still remember matrix or linear algebra, so if I give you a 20 by 20 matrix, and each position, each cells in this tells you the probability that one amino acid will turn into amino acid through evolution. And you have the frequency of amino acids in your protein. If I multiply this matrix to this frequency of amino acids, you get the frequency of amino acid after 1 unit of evolution. So basically this matrix can change one amino acid to another. And at the end you get like a composition of the new amino acids. So this model is called point accepted mutation or PAM. So PAM follow by numbers here. PAM1 means that you multiply the frequency of amino acid by this matrix 1 time. So this mimic one step of evolution. If you want to mimic like long-term evolution, like between distant species, you can multiply many metrics together. And the number of metrics that you multiply will mimic the evolutionary process for that many steps. If you multiply this metric twice, then the frequency that you get at the end is 2 steps of evolution. So if you see PAM2 is PAM1 times PAM1, so 2 steps of evolution. So you can imagine that for BLOSUM, is estimated using less conserved protein, like 45% or 60%, versus PAM, which mimic 1 step of evolution, which is very close, very short time period. So this two approach are good for different scale of evolution. And this is a rough kind of conversion. So for birth of 90, so birth of 90 means you construct the score based on protein with at least 90% similarity. So it's really conserved. This is roughly equal to using PAM 100 times. So you mimic the evolution 100 steps, 100 times, and you get something similar to a 90% conservation. If you further mimic the evolution for another 20 times, you get something similar to an 80% conservation. 160 times, you get something similar to a 60% conservation. So the more and more evolution you apply, the less and less conservation you get. So PAM is good if you have high sequence identity between your input and the database, because you are mimicking short-term evolution. But if your sequence are, you expect it to be very different from the database, like you don't expect your species to be there, then you can use boson, because boson allow you to search for similarity that are 60% conserved, 50% conserved, 45% conserved. And when you use BLAST for protein, there will be an option for metrics. Instead of the match and mismatch score, it will ask you to choose which boson or which PAM that is appropriate for your expected information. So not only the alignment score, like protein also have other techniques that can help us search for similar proteins. So the regular BLAST P, so BLAST protein, P for protein, it can assume that all amino acids are the same. It applies the same evolutionary models on substitution score to every amino acid position, right? Every position in the protein. But given your knowledge of protein structure and biochemistry, you know that protein have domains with specific function, like DNA binding domains, enzymatic domains, and different structure. So we know that not all amino acid positions evolve at the same rate or the same pattern. How do we take this into account when we search for similar protein? In BLAST for protein, if you want to consider this kind of pattern on the protein domain and other things, you have two options. If you know the pattern, they call it a phi BLAST, PHI BLAST. If you don't know the pattern, but you expect the protein to have some domain structure, some evolutionary pattern, then it's a psi BLAST. Let's see how these two works. So for pattern hidden initiated BLAST or phi BLAST, this means that you know the pattern and you can specify the pattern like this. So you know that the first residue must be LIVMOF, like you might know that this is important for binding or for certain activity. You know this must follow by a G and E. It can be then followed by any amino acid. X is anything. And then GA or S, and then another LIV or M, and then any amino acid, but the length has to be between 5 and 11. So we can specify very complex pattern in your search. And this means that you have to have a very confident knowledge of the domain that you are looking for. This is not usually what you would do. But let's say if you don't know the structure, if you don't know the domain, you want the BLAST algorithm to discover this for you. What it's going to try to calculate for you is called a position-specific scoring matrix. So that means each residue is going to get its own scoring scheme, not the same thing throughout. Something like this. Some position might be G all the time. Some position might be G most of the time, sometimes A. Some position might be A most of the time, and sometimes V, sometimes Y. So each position in the protein domain, because they have different function, they can evolve or they can change in a different profiles. How do we figure this out? How do we figure this out without knowing anything? So what psi BLAST does for us is it's going to start from your input sequence. You have a protein. You want to search for other protein. But currently, you don't know any pattern. You only have one single sequence. So it's going to do BLAST is going to do a single BLAST search, a vanilla BLAST P search. So we're going to get some hit from BLAST. And this is not going to be accurate yet because it assumes that all red amino acid position evolve in the same way. But once it does one round of BLAST, and we get more, we get some hit, we can do an alignment between these hits. And now we start to know, okay, the fifth position is mostly M, but sometimes V. This position is always R. This position is sometimes V, sometimes I. So with a single round of BLAST P, we start to get an idea of what are the amino acid composition at each location in the protein. So the program is going to construct the position-specific scoring from this hit that we get, and it's going to use that position-specific profile to run another search. So we will get a better hit this time because now we have a different scoring for different position. We get new hits. It's going to update the position-specific metrics and then run this again, multiple rounds. So this is a repeat until we refine the position-specific score and we get the final result. So with this, you can start with a single sequence, like you have no idea about the position-specific score, but with multiple rounds of searching, you can refine the position-specific profiles further and further until at the end, it should converge to the profile of your domain of interest. So this is an example. So I take a protein sequence that are new from a new indigenous plant in Thailand. We suspect it to be a CYP reductase, so it's a familiar protein. So we search this in NCBI, and we have something that very similar. So the coverage is almost 100%. So that means it's a full-length protein. But the identity is between 81% and 89%. So the actual species is not studied yet, not in the database, but we can get BLAST result to similar protein in different species. And in this case, in other species, people have already studied the function and they annotate it as cytochrome P450 reductase. So it's what we suspected, but now we can confirm it with a BLAST result. So this is one example of how we use BLAST for protein. Okay. In addition to BLAST, sometimes if you want to search for protein using the functional domains, the binding domain, certain domains, you can also use another tool called Interpros. And this will tells you that the protein that are similar, what kind of functional domains does it have. It can annotate your sequence with these domains as well. Okay, I think we are almost done. Okay, so sometimes, so we talk about a pure nucleotide alignment and a pure protein alignment. But sometimes you need application that can go back and forth between protein and nucleotide. And let's see what I mean by this. So in BLAST, in addition to nucleotide and protein BLAST, you also have these two options. Actually, there you have many more options, but let's talk about these two for now. So you have BLAST X and T-BLAST N. So in BLAST X, you will see in the description that this is a translated nucleotide aligned to protein. Why do we need this? So when we talk about nucleotide BLAST, we kind of assume that it's a genomic DNA. So genomic DNA, you have mutation. The mutation change any nucleotide to any nucleotide. But if you have a coding DNA in the coding region, we know that it has a codon structure. And any mutation, it can either change amino acid or it might not change the amino acid. So mutation in the coding DNA, not all positions are the same. And it has a protein, it can induce change at the protein level. So the best way to do sequence alignment for coding DNA is to translate them to protein first. and then do the alignment at the protein level, because we have more evolutionary information when aligning amino acids than aligning DNA. So that's why we use BLASTX if you have a coding DNA and you want to compare it to protein. But in another situation, you might do proteomics and you get a new protein. And you want to know if this protein is expressed in a different species. But unfortunately, the species that you are interested in might not be so well studied. Like it might not have a proteome. So even if you search with BLASTP, you're not going to get any hint because there is no proteome for that species in the database. But because RNA sequencing, we will learn it later on, but with RNA sequencing, it's much easier to study the species. So for that species, even if the proteome is not there, there might be a transcriptome there. And since the transcriptome is in the database, if you translate the protein from the transcript, then you can use those proteins as a database for your search. So T+N is when you have a new protein, but you don't expect that protein or that species to be in the database, but you expect that maybe an RNA sequencing might be there. So in this case, the translation of nucleotide will occur on the RNA database, and your protein input will be aligned, will be searched against that translated database. So it's a cool application that if you study non-model organism, you're going to like this. If you use model organism, then you can do the regular nucleotide and protein class. Okay, so I already mentioned the use case, but I summarize for you here. Okay, so let's end this session with kind of an overview of some more advanced alignment. So earlier what we talked about is called a one-to-one alignment. So we have one input and we align it to a different sequence in the database. But in actual application, we talked about this in the evolutionary lecture. Sometimes we want to do a genome-to-genome alignment. We want to consider all genes in human versus all genes in mouse, so that we can identify pairing of evolutionarily related genes. We don't want to do one-to-one at a time. We want to do all versus all search. So one genome versus another genome. So this requires a more sophisticated scoring criteria to figure out which gene in human correspond to which gene in mouse. Another setting is when we have more than one sequence. Let's say in this case, we have 4 sequences. There are many ways we can do the alignment. Right? We can split 4 sequences into 2 and 2, and then align them together, and then merge them at the end. There are many ways to do this. And in this case, what I can tell you, we are not going to worry about the actual algorithm. What I can tell you is dynamic programming is no longer working. Dynamic programming works on one-to-one sequence alignment because we only consider the sequence length. We can make the sequence shorter into a smaller problem. But if you have 5 or 10 sequences, there are so many ways that you can pair them together. For anyone who studied discrete mathematics, you will know how many ways to do it, but it's too large. You cannot keep breaking down the problem into smaller ones. It's going to grow very large. So dynamic programming is no longer possible. But the good news is there are different kinds of algorithms that can be used. And we might see this in different algorithms as well further down the course. But for multiple sequence alignment, where the possibility of pairing the sequence is growing larger and larger, we can do what's called a greedy algorithm. So greedy, as the English terms mean, you being greedy. This algorithm will try to optimize the next step without considering the actual kind of the whole thing. So for example, if the next nucleotide are T, then they want to align that together without caring that maybe after that you shouldn't align the T together because afterward there might be different nucleotides that do not align. But with greedy algorithm, if you only look ahead, maybe 1 step or 2 steps at a time, and then say that, okay, these are the best I can do right now, I'm going to follow it. So it's greedy in that sense. Or sometimes we do what's called a randomized algorithm. So let's say, okay, there are a million ways to align the sequence. We have no idea how to find the best one. So we will just pick 1,000 ways at random and then keep the best one. So it's randomized in the sense that we randomly make decisions and we keep track of the best one that we found. And sometimes we can guarantee that if we randomize and we sample enough, we can guarantee that we can get close to the best answer. Maybe not the best answer, but we can get close. So these are different class of algorithms that can help when the dynamic programming is not good enough. Okay. And if you're curious, these are like the output for the alignment. So we will have not the amino acid or nucleotide, but we start to have the dashes. So dashes here indicate the insertion deletion. And so we will see this in some assignment where you need to do the alignment. Okay, so this is it for our today class, focusing on sequence alignment. Like we learned about some algorithms like dynamic programming, especially, and we see many kind of consideration and details that link kind of how our goal from biology transform into parameters for sequence alignment. So if you have any question, you can ask, but for now I'm going to stop the recording.